It's my pleasure to introduce our first speaker of the session, Professor Hyungjin Kang, the director of the CAR T Cell Therapy Center of Seoul National University Hospital. He is the top researcher and clinician in the field of pediatric hematology, especially CAR T therapy. And for the details of his career, please refer to the program book. The title of his talk is Development of Chimeric Antigenic Receptor-T Cell Therapy. Professor Kang, please.
Thank you for the kind introduction. It's my honor to present here about my work for CAR T. Actually, I'm a pediatric oncology hematologist and not obstetrics, so it may be somewhat not your field, so I think. But actually, CAR T is the historical medicine who can cure patient with the cell engine, and its current technique and direction of this research method might be applied to your research. I hope so. And I will start my talk. Actually, the CAR T is firstly applied to the ALL, acute lymphoblastic leukemia in pediatrics. But although the survival was very good generally, but the survival for relapsed patient is very poor, maybe more 10% per second or so the relapse. And leukemia originated from lymphocyte stem cells. Actually, you are on the stem cells, I know, and during lymphogenesis, CD19 is expressed on the pro B cells, pre-B cells, immature B cell, and mature B cells. Malignancy derived from these lymphoid cells, such as ALL, lymphoblastic lymphoma, diffuse large B-cell lymphoma, and Burkitt lymphoma, in order of maturation, also express the CD19 antigens. Therefore, targeting CD19 is a promising strategy for treating B-cell malignancies. To eliminate cancer cells expressing specific antigen like CD19, CAR T therapy was developed. CAR T cells are engineered with a single chain antibody, not a double chain, it's single chain, and that recognizes the antigens, and then it couples with the intracellular T cell signaling domain, so it is called as chimera, which was different part from B cell antibody and T cell receptor. And the T cell transduced with the CAR constructor using a retro lentivirus enable them to recognize and kill the cancer cells. One of the most notable success of CAR T therapy was the case of Emily, a refractory ALL patient treated with CD19 CAR T developed at the University of UPenn. Her complete remission was published in NEJM, and Novartis subsequently commercialized the therapy and named it as a Kymriah, the T-cell. And this marked a breakthrough in science, medicine, and industry. Currently, 7 CAR T therapy has been approved by FDA and EMA and available on the market. In Korea, now we have 3 CAR Ts. One is reimbursed, and that is T-cell Kymriah, and the other 2, Yescarta and Carvykti is introduced in Korea, but not in the other CAR Ts are not interested yet. However, the history of CAR T began long before Kymriah. In 1987, long time ago, a Japanese group first proposed the concept. And the first CAR T experiment was conducted in 1989. So its efficacy limited by the well-known Weizmann Institute from Israel. So the IP is also diminished. With the advance in immunology, second generation CAR T cells were developed by incorporating co-stimulatory molecules and significantly improved the outcomes. Therapy like T-cell, break cell are example of second generation CAR T, and now third, fourth generation CAR T under development for further enhanced efficacy and reduce toxicity. Numerous early studies have been shown that the second generation CAR-T, CD19 CAR-T, with varying gene structure, either by antibody difference, costimulatory difference, and zeta viral difference, are also all effective in treating refractory and relapsed ALL. T-cell was introduced in the clinical practice in 2017, long time ago, already 8 years passed. But with very high cost. And unfortunately, at that time, it was not available in Korea. So, to address this gap, I explored a way to provide CAR-T therapy in patients in Korea. Fortunately, I discovered that Miltenyi Biotech offered an automated closed system capable of manufacturing CAR-T cells. After initiating discussion with colleagues at Miltenyi, they agreed to support the CD19 CAR-T study in Korea. And with funding from the Korean government research-driven hospital initiative, we established a production system, QA/QC protocol at our university hospital, and we successfully completed GMP-grade production, validation, and preclinical animal study, and received approval for a clinical trial targeting refractory relapsed ALL first time in Korea. We developed a point-of-care CAR-T production system with full qualification in our hospital, and we conducted GLP-level preclinical animal study of safety, efficacy, and distribution. And we developed a new integration analysis method named distinct sequencing, and performed analysis of CAR-T made in our hospital for IND filing. In 2022, we successfully treated our first patient. That year, 5 patients received CD19 CAR-T produced at our hospital. 2 of them, unfortunately, experienced CD19 negative relapse. All patients developed B-cell aplasia, indicating that the CAR-T cell were effectively targeted CD19 positive cells. And in 2022, T-cell, the Kymriah, received reimbursement approval in Korea. And we initially planned to stop our study. But the reimbursement criteria mirrored those of the Eliana study, excluding patients with less than 5% bone marrow blast, isolated extramedullary relapse, MRD relapse, or complete remission after chemotherapy. It was a study by Novartis, and it is the basis for the approval of Kymriah. But in real-world data suggests that patients with extramedullary relapse can also benefit from CAR-T therapy. They can be cured by the CAR-T therapy. And in our own trial, one patient with extramedullary relapse experienced the complete resolution of the mass following CAR-T treatment. Therefore, we have continued our trial to provide the CD19 CAR-T therapy to patients who meet clinical indications but are not covered by our insurance system. So far, we have treated 8 patients with our CD19 CAR-T therapy. Among them, 6 are alive. 2 patients received the transplant after CAR-T. One lost BCA aplasia, and another required etoposide, a kind of chemotherapy to manage cytokine release syndrome, cytokine storms. And this slide shows the case of a 7-year-old patient whose leukemia relapsed multiple times, eventually presenting as extramedullary relapse. After receiving our CD19 CAR-T therapy, the mass completely disappeared. As the first IA for CAR-T in Korea, our work was selected as one of the top 10 scientific news stories in our country, and the Ministry of Health and Welfare recognized our study as a major achievement, and I was honored to receive a prize from the ministry. Although CD19 CAR-T therapy have shown promising result and is now in part of clinical practice, a significant challenge remains, particularly about the severe adverse event. Known adverse effects include the cytokine storm, the theoretical CAR-T cell transforming into leukemia, and on-target optimal effects such as B-cell aplasia. 2 cases of T-cell lymphoma associated with BCMA CAR-T therapy have been reported. Although the exact pathogenesis remain unclear, first they think that it might be incorporated near the oncogene, but it was not. But after the reviewing relevant clinical data, the US FDA and medical experts concluded that CAR-T therapy should continue, as the potential risk of malignancies outweighed by its anticipated therapeutic benefits. And among the adverse effects, off-target and unexpected effects are the most concerning. In a clinical trial by Juno Therapeutics using CD90 CAR-T with a CD28 co-stimulatory domain for adult leukemia, 5 consecutive deaths occurred due to brain edema, but the exact cause remains unclear. And to improve the safety, mechanisms such as the suicide gene and on-off switch have been recently introduced to improve the safety. They can stop the CAR-T when there is some adverse effect occurs with this switch. And another unmet need of CAR-T is that its effect is not perfect. In patients treated with Kymriah, relapse occurred in about half between CD19 positive and CD19 negative. CD19 negative relapse often due to antigen escape under CAR-T pressure and is associated with significantly poor outcomes. Cancers are very smart and they can avoid the CAR-T with decreasing the antigen level in their cell surface and escape the CAR-T treatment. And to address this bispecific CAR-T targeting both CD19 and another one like CD22 has been developed. And this includes co-administration, bisystronic, and co-transduction and tandem. Many ways are under development to increase the efficacy by two antigen targetings. And many techniques are under development to enhance CAR-T function, especially in the harsh microenvironment of the solid tumors. Recent success include clouding CAR-T in gastrointestinal cancers by China, and GD CAR-T by brainstem glioma, which was fatal but can save the life by Stanford, and the GD CAR-T in neuroblastoma in Italian groups. And many next-generation molecular platforms for CAR-Ts are under development to improve efficacy and to avoid toxicity. And enhancement strategy for CAR-T technology are under development that are already in clinical trial. You can see the bold, and this is all in clinical trial now. And made CAR-T with good T-cell because it's biologic product is another way. In lymphoma study, DNA methylation signature of harvested T-cell before CAR-T manufacture, named EpiCAR-T, was associated with survival. And our group reanalyzed the result and then found that EpiCAR-T signature of harvested T-cell malignant harvested T-cell predominantly reflect the naive T-cell subset. Means that CAR-T made from naive T-cell might be better than the memory T-cells. And the reduction of manufacture cost and time is one of the main directions. To make current autologous CAR-T, leukapheresis is performed to get mononuclear cells, and 2 T-cells were selected, activated, and transduced with virus vector encoding CAR. And after expansion, CAR-T was harvested and infused to patient. But the real manufacture process is very complex and also need many qualification tests. Compared with the previous medication, autologous CAR-T is a personalized manufacturing drug, need qualification for each class. So the manufacturing cost is very high. As a result, financial toxicity is a big concern about this new high-tech treatment. Not only the CAR-T, but the other stem cell-based gene therapy. The high cost is almost anywhere. It is very big problem. And one way to reduce cost is the manufacturing innovation. Conventional manufacturing methods need a lot of manpower and equipment. And automated closed systems such as Prodigy have been introduced for easy manufacture of CAR-T and to reduce cost. And manufacturing innovation such as one-day CAR-T production is a way to reduce cost. And recently, Novartis introduced this system and started the clinical trials. And using non-viral vector instead of viral vector is a way to reduce cost because viral vector is very expensive. And the viral vector can be infused in the patient directly and made CAR-T in the body, named in vivo CAR-T. And this in vivo CAR-T is an area of interest in clinical trial are now ongoing with this technique. We think it can dramatically reduce the cost because virus itself can be a drug and it is not a cell drug and virus can be a drug and treat patient with a very low cost compared to conventional CAR-T. And as current recently published in Science, making in vivo CAR-T using RNA and an antibody conjugated nanoparticle is a promising way to overcome safety issue of the cost. And another way to reduce cost is the production in the point-of-care medical centers. By point-of-care production like us, we can reduce cost for transport, which was also very expensive because Novartis used many money for cell chains from the hospital harvest and to go to the US and made CAR-T and transport to our hospital and infuse. So during this, they made a cell chain and it's very expensive one. And another way is the production of CAR-T in country where drug development and manufacture is significantly cheaper. It was news in the Nature that recently cheaper domestic CD19 CAR-T was approved and in the market in India. And another way is to make off-the-shelf third-party CAR-T and CAR-NK, which was already made, and if patients need it, they can use it as like a drug. And gene editing technique has been developed and introduced in the CAR-T field to remove alloreactive T cells because alloreactive is very big hurdles between patient and donors. So it is important to remove T cell receptor and HLA before CAR-T infusion to use the third-party CAR-T. Recently, TALEN, CRISPR, and base editing like technique was introduced and also in clinical trial with good success. There is a clinical study using third-party TCR, removed CAR-T from healthy donor, and this kind of off-the-shelf CAR-T can be used when necessary. Under the off-the-shelf method, it uses CAR-NK because NK cell we can treat it for on a third party because it has a low alloreactivity, so we can use NK instead of T, although the duration of treatment is very short. So there is a pro and cons using NKs. And the beautiful data was published with the HLA mismatched anti-CD19 CAR-NK from cord blood by MD Anderson groups. A further challenge is the complexity and high cost of manufacturing. The process involves leukapheresis to collect, slide. And I will now talk about the emerging indication of CD19 CAR-T in ALL and the attempt to use the CD19 CAR-T therapy in earlier line treatment. Although early data from T-cell Eliana study for refractory relapsed ALL showed promising result and led to its history approval for ALL, it was early data showing from 10% to about 60%. But long-term data shows not so satisfactory, only 35% event-free survival. As you know, axi-cel and liso-cel has been approved as a second-line therapy for DLBCL in adult, and similar efforts are ongoing for pediatric ALL. They are moved from third line to second line and showed efficacy and are now sold in the market. but not the Kymriah. According to long-term follow-up data in ALL, patients who achieved hematologic CR before CAR-T means the low tumor burden showed better survivors than others. It means that you also have experience of killing a cell, and you have a target cell, and the T cell ratio might be very important to kill the cancer cells, and it is very reasonable that with low tumor burden can be very important to enhance the efficacy. But CD19 CAR-T was less effective when CD19 antigen burden was very low before infusion. Means that CD9 antigen or B cell is necessary for the proliferation of T cell, because T cell is proliferate when they met the antigens. So, considering this finding, patients with a low disease burden and sufficient CD19 positive normal B cell are the ideal candidate for CD19 CAR-T. The best effect of CD19 CAR-T might be seen in patients with hematologic CR, means low blast, less than 5%, and normal B cell recovery. In this context, patients indicated for transplant who are in hematologic CR may be the best candidate for CD19 CAR-T. While the allogeneic transplant remains a curative option, it can be lead to a long-term toxicity such as infertility, almost in all, and then lung fibrosis, and many patients are succumb to lung transplant, and so on. To improve CD19 CAR-T outcome and potentially avoid the transplant, a clinical trial is ongoing in pediatric leukemia with MRD positivity after consolidation chemo, which is very high risk of relapse. In Korea, the past chairman Kon Hee-ri of Samsung Company donated big fund when he passed away in 2021 to support pediatric cancer and rare disease patients nationwide. With this fund, we can launch a nationwide multi-center CD19 CAR-T trial for pediatric ALL patients indicated for allotransplant, aiming to improve the survival and potentially avoid transplant in 50 patients. CD19 CAR-T cells are manufactured at our hospital GMP facility and then delivered to the hospital where the patient received the CD19 CAR-T. Here are some cases of our study. A patient with NGS-MRD positive means that there is a minimal leukemia left after the chemotherapy. It is very poor risk factors. And he received the CAR-T and achieved the NGS-MRD negativity and underwent transplant and live now. CD19 CAR-T before transplant may improve outcomes. And some patients with very high risk of relapse who achieved the NGS-MRD CR through the chemotherapy received our CD19 CAR-T and were able to avoid the transplant. It was they did not receive transplant and only received CAR-T and survived. Last year, the current chairman of Samsung visited our hospital to attend the memorial ceremony for the past chairman. And during the visit, a patient who have received CAR-T and avoided transplant expressed their gratitude. And our current CAR-T study is attracting a lot of media attention in Korea. When we think about the development sequence of T-cell Kymriah, UPenn, University of Pennsylvania first reported 2 cases, including Emily, 1 failed, and they performed investigator-initiated clinical trial and in 30 patients and transferred technology to Novartis, and Novartis performed a sponsor-initiated clinical trial in 75 patients and got approval. This is same for Yescarta, Brainage. After the success of IIT, they become drug from academia to industry. Currently, about 1,000 CAR-T clinical trials are ongoing worldwide. But in Korea, we are somewhat behind the global trend in the field of CAR-T, and the number of studies is small. Among them, our study is one and recently two investigator-initiated study. As human validation is necessary for CAR-T to make chimeric drug to develop a new cell and gene therapy, small-scale IIT, academic trial, and then SIT, the industry trial, commercial trial, and then commercialization is the best sequence. Academic trial is under relatively favorable regulation and fast and optimal way to apply new treatment to human, and the relatively little economic damage even after failure. Our work for CD19 CAR-T was probably one that can cure sick child and validate CAR-T development platform of our hospital composed of a GMP Animal Center and Clinical Trial Center to boom up new CAR-T trial in Korea. We hope to validate novel idea about CAR-T for many kinds of disease in human with investigator initial trial and academic trial, and develop a new CAR-T and transfer it to industry sector for commercialization. CAR-T is a technology to connect antibody and cells, and we can give logic, genetic logic to cells with gene modification. And when I explain current commercialized CAR-T, like T-cell Kymriah, I said it's like the first generation cellular phones. We could call without line, but it was very expensive. As it become a smartphone in your hand, CAR-T will be developed and become smart and popular drug. In future, I hope many new CAR-T will be developed and hope to cure many diseases. I thank to my team and colleague and Milteni for their devotion to make CAR-T academic trial in our hospital. Thank you for your attention.